So we are delighted this morning to have Patience Mthunzi-Kufa with us. She is the lead for biophotonics research here in South Africa at the Council for Scientific and Industrial Research, which is South Africa's Central Research and Development Institute. She's using lasers to characterize and treat disease, looking from embryonic stem tissues to HIV to cancer. And she will talk to us today on biophotonics and artificial intelligence for improved diagnostics. So thanks so much for joining us remotely, Patience, and we look forward to hearing your work. Thank you very much.
Hey, thanks, Adriana, for the introduction, and good afternoon, or rather good morning, everyone. I'm joining you this morning from the Union Buildings as I've come here for the reviews of the national orders, because I belong to that committee as well. So that's why I couldn't join you in person. So I'm sitting in someone's office. I hope that I will be with you for the duration of the talk, that there won't be any interruptions or disruptions of my internet connectivity. And if there is, I do apologize in advance. I do have, though, a stable source of internet. I brought my dongle with me, so let's hope that we stick together until the end of the talk. My laptop does have a tendency of freezing from time to time. So if that happens, I will just move from presentation mode to just a slide presentation, and hopefully that will not affect the resolution too much. But thanks, Adriana, for the invitation, and I look forward to participating and also to hearing what the audience has to say. My talk is not going to be mainly technical. I will be touching on the applications of biophotonics in health, in pharmaceuticals, as well as in agriculture. I will share some of our lab data because we've done preliminary tests on all of the aspects that I'm going to be talking about this morning, and I can then share with you whatever it is that we have. So I will try to go on presentation mode or slide mode now, and hopefully, you know, things behave until I get to the end. So if it's okay, Adriana, I'd like to start now. Is that fine?
Thanks. Go ahead, Patience. I don't know if you can hear me from the back, but if you can,
thanks.
You're all set.
Okay, so as in the introduction, Adriana did mention that I am from the Council for Scientific and Industrial Research, and this is the CSIR campus. And I mean, this is a very big organization. We do research across the spectrum. And just, you know, as a matter of introduction, I'd like to tell the audience that it's not only photonics that we are focusing on at the CSIR, but there's a lot of work that's being done on the energy sector, a lot of work on the agricultural space. There's a big IT department. We've got smart mobility, smart places, and we've got mining research. We also have manufacturing research, and the manufacturing cluster is where I reside, and it's where I belong in. And our building is somewhere at the back there of the campus. So this is the entire campus, but our building is at the back there. So if there is people that have got appetite for collaboration, please do holler on us. Please do visit our website and let us know. We are always happy to take hands with other researchers and to collaborate. So I'd like to zoom into the biophotonics work that's being done within the Photonics Center at the CSIR. And right now we belong to the CSIR, but the idea is to actually spin off as a company. And what we call ourselves, if we do spin off, is Ukukhanya Diagnostics. And this would be our brand name. Ukukhanya is a Zulu word for light. So if you use it as a verb, it means to shine. But if you use it as a noun, it means brightness. So what it basically means is that we are able to shine light on any molecule, be it chemical molecules or biomolecules, and be able to inform what exactly the composition of that particular entity is, whether it's a cell or it's, you know, it's a tablet or whatever the case is. So we are using light to interrogate molecules in order to acquire a lot of information about those molecules, and that's how we do diagnostics. We are in a space that is quite unique in the sense that the composition of the team, we are a team of 18 people. The composition of the team, the laboratories themselves, as well as the research that we do, it cuts across the spectrum. So we are in the photonic space, but we have a good understanding of microorganisms, be it viruses, bacteria, or whatever tissue you would like to throw at us, because we've got a lot of virologists in the group. But not only that, we are also in the space of medical diagnostics. So if you look at this type of fashion or type of combination, it's quite unique in that sense. And that's what sets us apart both locally as well as globally. The first technology I would like to take the audience through is that one of a smartphone diagnostic. Why a smartphone diagnostic? I'll explain just now. If you look at our country in South Africa, I'm currently in Pretoria, which is about 6 hours or so away from KwaZulu-Natal, okay? So the dynamics in South Africa is that, you know, there's places where people go to a clinic and they have to access it via a mobile van, you know, or containers such as the ones that you're seeing here. This is an HIV clinic in a rural area in KwaZulu-Natal, okay, which, as I said, is about 6 hours away. But there are spaces like this not too far from, you know, the cities here in Pretoria. Now, for people in these areas, it's quite sad that sometimes they have to walk long distances to access this mobile van to come and do diagnostics. And we know how important it is that a patient gets diagnosed quickly, because if you get diagnosed quickly, you can actually avoid disease progression, and you can actually start getting treatment for whatever it is you suffer from. Now, in the case of HIV, you need to catch it quickly before it actually progresses to becoming AIDS and killing the patient. And nowadays, we've got an efficient system through ARVs, antiretrovirals, that patients can take. But the problem here is that sometimes a patient gets here and the van has already left. So there's nothing that's going to courier their sample to a laboratory in the city that can then inform the patient what is it that they have. If they do have HIV, has it progressed? At what stage do they need to start ARVs, etc.? It becomes a problem because then it hampers on the early diagnostics. So patients have to be delayed in terms of diagnostics. And I'm just mentioning the case of HIV here, but same for TB, same for COVID, as well as for non-communicable diseases. So what my team and I set and thought of as a solution or a potential solution to this problem was to take a laboratory like the one you see here on the picture on the left to the people through a smartphone. So, you know, I make it sound like it's a 1, 2, 3 type of thing and it's easy like that, but we have built hardware which is novel through optics. So there's a lot of optics that have gone into building this device. And then we have put that hardware into a phone. And we've also made a lot of novel software through apps and programs that can then help us to read the sample that you see here using this particular phone. So we are putting both the novel as well as the the novel hardware as well as the novel software into a phone to be able to generate something like a lab that a patient can use in their point of care. And point of care can be anywhere, including a patient's home. So we have done that. And I must say the technology has progressed quite high in the technology readiness level. It's currently at technology readiness level 7 to 8, because we've now generated a lot of apps and we are testing it using real samples. And some of the software as well as hardware that's been put here is being protected via patents. Because I'm talking to a team that's working with biophotons and they understand how, you know, these photons can be illuminated in the body of an animal and they can actually emit light. I will mention some of the technologies that we have come up with in the team, and that includes how we prepare our samples. So in this particular slide, I'm actually sharing some of the methods, and that's because I realize that sometimes, because, you know, I'm talking on work that's being protected, I then deprive the audience an opportunity to see what our optical setups look like. So in our facility, as I said, we have a facility to do cell culturing, and this is a typical sample preparation that we would have. We are able to run ELISAs, and we are able to also immobilize antibodies for HIV, for TB, as well as for COVID on particular surfaces, and then perform optical experiments on them. The first setup that I'm showing you there is the photonic crystal biosensing setup, where you can actually see more or less how the setup has been put up. And then we have one for local surface plasmon resonance. And the SPR setup also looks something like that. We've got numerous lasers as well as LEDs that are the heart of these setups. And then we have that smartphone biosensing setup that I spoke about. And this is just an example of what one would expect to be in a setup like this. And then we also have a setup that does low laser therapy, or low-level laser therapy. And this we actually use to actually analyze some of the COVID samples, as well as the HIV samples. But I think in this presentation, I've got some results for the COVID samples. So this is just giving you a flavor of the type of equipment and the type of setups that we have. So when it comes to some of the results, you can see that the preliminary results show that our experiments actually work. If you look, you compare what happens with the photonic crystal data, you can see that the PBS, which is just a reference sample, the trend there is slightly different in terms of the angle shift compared to that D6 sample. Unfortunately, I couldn't show these as individual slides so that the audience can read them properly, but I think the audience gets the idea there with the angle shifting. And then when it comes to LSPR as well, you can see that our preliminary data is showing that, our experiments actually do work. And then when it comes to the smartphone one as well, again, that's a very bad slide because everything is crammed up. But the idea was just to show preliminary data that's looking positive. And when I show you now here, the viability as a result of treating the samples that are infected with COVID as opposed to the ones that are not affected, you can see again that there's positive data there. Some of this work has been already published in papers. So if you would like more details, you can just request for some of our journal articles or just go online in the research space and you can get them. So that would be my message this afternoon about the smartphone work. I'd like to also now look into another technology that bears a lot of potential, and that's the LAMP Arduino for PCR. What do we mean by this? Most of the diagnostics, be it HIV diagnostics, TB diagnostics, or even COVID diagnostics, are being run via a process called polymerase chain reaction, and that's a PCR. This is like a gold standard method of doing diagnostics when it comes to most of the communicable diseases. And I think the audience would agree that following COVID, most of the tests or most of the people came across this PCR technology because it was being spoken about in a number of cases where tests were being done and, reports were being made as to how many COVID positive samples have been picked up. So PCR was the gold standard, it's accurate. But the problem here with PCR is that it's quite tedious to set up and it's time consuming, and it also is quite laborious in the sense that you have to have a trained technician to run the technology in the lab. It's very expensive as well. So what my team and I had a look at was a possibility to can maybe replace this PCR. And we came up with this handheld loop amplification, isothermal amplification Arduino system that we are actually using to replace the PCR. So what we did was to teach the LAMP Arduino all these things here. So we had a simulation where we are actually mimicking how a PCR works, but then we put all of this in the Arduino that I've just shown you now. If you look at this picture, you can see that there's a camera there. Sorry, I'm just hiding the electronics for protecting some of the IP, but there's a camera in front of the Arduino that is currently looking at the keyboard, and that's what you are seeing there. So if the keyboard were the sample, that's what you'd be picking picking up there. And that's the hardware of the Arduino that you've just seen now. But on the flip side, we also have generated the software where the welcome screen on the phone would be looking something like this, for example, where you have the opportunity to incorporate information, simulate, and then maybe also get some help if you are stuck in the app. You can read different diseases like TB, HIV, COVID, and you can also have the exit button. But let's say, for example, in this case where a TB sample was read, you incorporate the sample, and as it's being read, you then get a trend. So this is just to give you an idea of how a typical system works. So this would be how the Arduino that's being put on the smartphone or that's been read using the smartphone actually looks like in terms of software. There's a lot of machine learning work that the team is also working on. And here, really, what we are doing is we are working with repositories that keep data for different diseases. So you would, for example, get a spectrum for SARS-CoV-2. And then you would process the spectrum, and you would, through machine learning algorithm, get to a point where you have classified it. So as I said, we get our sequences from repositories, and some of them come in a form of spectra. And just by looking at the results, you can see that we have used 3 different models, and we have tested the level of accuracy of each model. And you can see that, especially with the ones that take the longest, the level of accuracy is almost close to 100%. And for each of these models, we actually tested 62 Raman spectroscopy samples. So this is another type of preliminary data that we are picking up using our machine learning from optics data that has been generated in the lab for different diseases. In this particular case, it was just SARS-CoV-2. Moving on, we can see that we have achieved quick and automated diagnostics of diseases, and the system is efficient in extracting valuable information from spectral data, and it is, improved in terms of accuracy and also in terms of the turnaround time, because some of these gold standard tests take long to actually run. You can take anything between 48 to 72 hours to getting your results. Whereas in our cases, when you do a Raman spectra of a particular sample, within 15 minutes, you've got a complete picture. And our future perspective would just be, looking into improving our algorithms as well as, to resolve the challenges of the limited data that we are currently facing. In another experiment, the group is working on designing microfluidic chips. And this is what we've come up with so far using the 3D printer that we have. So we've got a 3D printer that allows us to do printing of microfluidic chips. And what I like about these technologies that I'm talking about, you can integrate them into each other. You can miniaturize the integrated technologies and get away with just having a handheld device that can be used for diagnostics purposes. So this is just the flavor of the research that's happening within our space at the CSIR. The audience is more than welcome to contact me if you would like to do a site visit. We are more than happy to take you through our technologies. And as I said earlier, we are happy to also collaborate. Another big issue that we have seen in South Africa and in Africa in total is that of substandard medication in Africa. We spoke to the CEO of Kiara Health, who visited our facility some time ago and said to us, your technologies do beautiful things, but I'll tell you one thing, if your technologies can actually pick up substandard medication, that would be a very good thing in South Africa. It would be quite impactful in the pharmaceuticals in South Africa and in Africa. And that's because in all the medication that we get, 1 out of 10 of those medical products is actually a substandard or falsified product. It's a big problem. So you get in other countries in Africa, like in this region here in Abidjan, a lady sitting and demonstrating and selling actually all these pharmaceuticals in a market like this. Now, I don't need to emphasize the point that pharmaceuticals are just like food. You can't just store them anywhere. We don't know if where this lady is seated, there's a shade. We don't know if there's specific conditions to preserve the active pharmaceutical ingredient of these products, but this lady is selling them. People are coming to consume. And sometimes people are consuming medication that has already expired or medication that's actually going to do them harm than good. So we are trying to solve problems like this. And we can see that the market for this type of work is actually scaled at around 200 billion of dollars. And 42% of cases are found in Africa, where we find that 250,000 children actually die on a yearly basis due to substandard medication for malaria or pneumonia or even TB. So it's a big problem. And common counterfeits are found in ARVs, and that's why in some of our work in the CSIR Biophotonics team, we have actually started screening ARVs, and we've come up with sensors, novel sensors that actually help us to enhance the signal for picking up ARVs using our spectroscopic techniques. But you can also see that antibiotics are also in the list of some of the very common counterfeits, as well as anti-malaria products. I hope I'm still with you, Adrian. You know, sometimes you talk to people because it's quiet, you think you're still talking to an audience, but I'm talking to myself. If I can just get confirmation.
Yeah, it's okay. Hear you fine?
I don't know if you're saying upstairs.
Yeah.
Okay. All right. Thanks for the confirmation, team. All right. So here what we did was the gold standard medication that has been used to date for picking up any type of chemicals when it comes to pharmaceuticals, and that's being used for doing the QC, quality control in pharmaceutical industries, is HPLC, right? High pressure liquid chromatography, as well as mass spectroscopy. And again, this is a laboratory-based technique. It is highly sensitive. It is good, and it's been relied upon for years. It's got high maintenance, as you can expect, and as I said, the turnaround time is very long, and it's incompatible with a lot of our IR systems. So what we have done is, in the Biophotonics lab that I lead, we've got this state-of-the-art Raman spectroscopic system that is currently being used to do exactly the work that's being used, that's being done by HPLC. It is highly sensitive. There's ease of use, and it can be digitized. There's low maintenance. There's about 15 to 30 minutes, as I mentioned earlier, turnaround time, and it's compatible with a lot of 4IR as well as machine learning technologies. So, we want to actually come up with a multi-screening device that's portable enough, that can be put in all entry ports, whether it's seaport or airport in our country, that will be able to do bulk screening of medication as it comes in the country before it gets dispatched. So that's how we are actually wanting to make an impact in assessing counterfeits. So yeah, I'm just sharing a little bit of our lab data. You can see here that I've got that 3-in-1 antiretroviral medication, and we've run a Raman spectroscopy of it. And you can see the medication in its pure form, and then we decided to spike it, and you can definitely see that there's a change in the preliminary data. There's a change in the spectrum. And we are getting results like this. It's quite exciting. So we would like to pursue the research and get to a point where we are actually formulating those devices, as I said. Another big industry when it comes to this kind of work is that comes from the indigenous medication, and CSIR has got a big indigenous systems indigenous knowledge systems group that's working on these medications. Usually they do have the sample, they do have the medication, but they don't really know what it looks like molecular wise. So they've given us samples and we've run it on our system, and you can see there that we've got 2 trends for gallic acid and 1 for quercetin, where we've actually characterized and informed the colleagues what it actually entails or what it actually contains molecular wise. So we managed to come up with the molecular fingerprint of those 2 samples that we were given. And then the other thing is, as I mentioned, we do make novel chemical sensors that help us to also improve on the signal. If you look at these 2 samples here, we just ran it without using silver nanoparticles, and then we use silver nanoparticles to coat our surface of the actual sample chamber, and you can see that the actual trend has been improved from what you are seeing there without the coating using silver nanoparticles. Right. I've already started mentioning these, but maybe delving deeper into that, in Africa and in South Africa, there's a lot of people who still consult with traditional doctors. And because we just want to keep the communities safe, we have now also started working through the Indigenous Knowledge Systems Group. We've started acquiring some of these medications to just come and, quantify and characterize them molecularly and tell this lady what it actually contains, because she can maybe recommend it for a particular sickness, but in most instances they're not. She doesn't even know what it actually contains in terms of ingredients. So just to keep things safe, we are now taking hands with them to be able to tell exactly what's in their medication so that the patients know what they are taking. And if people are allergic, then they can also be advised to avoid that particular type of ingredient. So that's just some of the work that we are pushing from our end. And then maybe lastly, let me touch on an area that we've just recently started working on, and that's artificial intelligence for agriculture. And really the applications here is for food safety as well as food security. We would like to apply photonics and, the recent technologies like AI as well as machine learning for making sure that we've got food and the food is safe for human consumption. Because in a lot of instances, there's also pesticides that are being used to keep away pests, but then you find that they are toxic as a food item to people. So we want to use our technologies to diagnose if the food is safe. And the reason why we are also doing this work is that right now our country is being hampered by load shedding, but very close to load shedding and maybe the lack of electricity, sufficient electricity at this point, we are just using foresight to actually tell that in future there's going to be shortage of water. There's already issues with water in a lot of our provinces in the country, but it's gonna get worse. And should that happen, then there's going to be a lot of challenges in actually getting the food. So we are trying to maybe get to a point where we are coming up with solutions for food safety and food security before we get to that dire situation. So, everyday farms produce thousands of data points on temperature, on the soil, the condition of the soil, the usage of water, and weather conditions, etc. Right? And with the help of AI as well as machine learning models, this data is leveraged in a real time for obtaining useful insights like choosing the right time to sow the seeds, determining the crop choices, and then hybrid seed choices, and generate more yields and the like. All right. So we can use these technologies to, make a lot of good choices that will set us ahead in terms of getting the crops. AI systems are also helping to improve the overall harvest quality as well as accuracy, and this is known as precision agriculture. And precision agriculture really relies on a lot of photonics technologies, be it your spectroscopy, be it your Raman spectroscopy, or different types of photonics technologies you can actually apply for precision agriculture. AI also helps in detecting diseases in plants, which means we can help to preserve the actual condition of plants so that they can then produce that crop that we want. You know, sometimes we can also get away with pests without using pesticides, and also, you know, be able to get the best quality in terms of nutrition of the products that we are farming. Finally, the AI sensors can detect and target weeds and can decide, you know, which herbicide to apply within the region. So, you know, there's a lot that you can do with machine learning as well as artificial intelligence in the agricultural space. And this is where the team and myself are now actually trying to venture. And the three different categories of agriculture is the pre-harvesting stage, the harvesting, as well as the post-harvesting. And the nice thing about machine learning as well as AI is that you can apply these technologies across these important stages in agriculture and be able to inform different subcategories of these particular categories. I think this is where my talk ends for this afternoon. It was just giving the audience a type of flavor of the kind of work that is being done within the biophotonics space at the CSIR. What we actually want to come up with is this assured criteria that has been put by the WHO to say that the diagnostics that are needing to be applied at point of care need to be affordable, they need to be sensitive, specific, they need to be user-friendly, they need to be robust, but they also need to be equipment-free so that you can use them away of the lab, and they need to be able to be deliverable to the end user, and they need to have that kind of ease of use and application, because it's not always going to be the case that if you are at let's say a rural area, you will be able to find a lab or someone that can actually interpret the results in the lab. So this is my team. You can see I think there's, aside from myself, it's nine ladies and 9 guys. So there's a balance in terms of the gender. And, you know, as I said, you know, I'm looking at different disciplines in terms of their training and their qualification. We were in our microfluidics lab here. We also do some of our molecular biology work in this lab. And yeah, I would like to thank you for your attention.
Thank you so much, Patience. We are really grateful for your time. I think this is extremely important work you're doing. So thanks so much for sharing it with us. We've talked a lot about the biophysics and the biochemistry and the experimental setups around biophotons. But I think the value of your contribution today has been to bring us back to the end user, the people in the communities, and the potential improvements in healthcare and really improvements in people's lives that can be achieved with the amazing innovations you guys are developing. And the application of the research to the people at the end of the day. So thanks very much for your time. We appreciate it. I think you can see us.
Yes, I can.
Looks like it. Okay, great. So let me make you bigger here. And let's open for questions. Are there any questions? Sorry, let me quickly. There we go.
Yeah, I have a question. I'm off camera. Yeah, I'm over here. Okay, so take for the talk. The adulteration of like drugs is something that we often see in like traditional Chinese medicine, traditional Indian medicine. But what happens is you get caught between, like, in catch up by the adulterators, who they come up with more and more sophisticated ways to hide how they process and adulterate the herbs that they're trying to sell. And I wondered if you had considered stuff like activity-based assays to determine false drugs, because we found that with some popular Chinese medicines and popular Indian medicines, they are so good at hiding that the drugs are fake that the only way you can tell is with activity assays. And I wondered if you'd considered that in your work.
Okay, thanks. That's a very good question, actually, and very good suggestion in trying those active assays in order to quantify and qualify the drugs. We have not tried it in our space. So as I said, we are mainly using spectroscopy to come up with the molecular, fingerprint of the chemicals, and then we can take it from there. But I mean, it's not to say you cannot try that, because, some of the molecules maybe you cannot pick up, but so far the preliminary data is showing to be good using Raman spectroscopy. And as I said, development of those sensors that actually also improve the signal of the Raman data. So yeah, we've not tried that, but it's a very good suggestion. It's something I will keep in mind.
I think it's a big problem with wine as well. Apparently they do it adulterated, or like they can mimic certain wineries, and you can't really tell them apart using traditional counterfeiting detection methods. So it's like you're always playing catch up with the people trying to falsify things, to the point where you can only see the actual activity of the drug. So it's an interesting catch-up game with the perpetrators that we've seen. Actually, and with Chinese medicine, where some of the export herbs can be very expensive. They're very good at hiding the counterfeitness. It's an interesting area of something that we hadn't really thought of before.
Yeah, definitely noted that. As I said, very interesting, and it's worth looking into.
Yeah, thank you.
Other questions?
Yeah. When it comes to your Raman fingerprinting database, I'm wondering, is there any reason why you're not using many of the available Raman fingerprint databases out there?
Sorry, using what?
So you have Raman fingerprint databases that you can use already. So what's the advantage of making them up yourself?
Oh, okay. So the aim here is to actually be able to come up with specific sensors that will pick up specific ingredients. And that's because we are looking at treatment of diseases in Africa, right? You might find that some of the biological molecules in, HIV, for example, let's take HIV. In subtype C infections cannot be picked up with, markers from outside of subtype C. Let's say in America, it's predominantly subtype B, and in the UK, it's subtype B in terms of infections. So we are trying to come up with specific ways of actually specifying the way we pick up molecules in the African context or in the South African context. And that's why we are going by it the way we are. We want to make sure that we address specifics first, and then obviously our technologies can be applied globally. But for now, when we are still focusing on what the scenario is in Africa, this is the approach we are taking. Then we can come up with specific sensors that address molecules that we are picking up in diseases as well as in the drugs. The ARVs that are used in Africa are not necessarily the same with the ARVs that are used elsewhere in the world. So we need to be able to adapt our systems to actually communicate with what we have in the country.
Further questions? Nothing further? Yeah, well, I know you have another meeting coming up, so thanks so much for making the time to share with us, Patience. We'll be in touch about the work that we do collaboratively after this. Yeah, as a South African, I'm really proud to hear about the work going on at the CSIR in your group, and I'm sure our international guests in particular have been excited to hear about the South African and the African context in biophotonics. Thank you, Patience.
Thanks, Adriana.
Hope to meet in person soon.
Thank you. Bye-bye.
Bye.